afternoon. Thank you for the opportunity. I'm Moyra Machado Portilho. I'm a PhD student from Viral Hepatitis Laboratory in Rio de Janeiro, Brazil. My presentation is about the usefulness of saliva samples for hepatitis B virus DNA detection. It is estimated that more than 1/3 of the world's population has been infected with the hepatitis B virus. According to this map from the World Health Organization 2014, the world is divided in 3 regions of HBsAg prevalence, being the north region of Brazil and countries from Asia and Africa, the most prevalent areas. About 5% of the population are chronic carriers of HBV, and nearly 25% of all carriers develop serious liver disease, such as cirrhosis and hepatocellular carcinoma. And HBV infections cause more than 1 million deaths every year. Concerning the virus, it is from HepDNA viridae family and HepDNA genus. The infectious particle, that is also called virion or DNA particle, is composed by a nucleocapsid, an icosahedric nucleocapsid, covered by a lipoprotein envelope, and in its interior there is the DNA and the polymerase complex. The DNA is circular and double-stranded, composed by 3,200 base pairs, and composed by 4 open reading frames that will codify the viral proteins. The infection causes a serological response in infected people that are related to clinical and cellular events. And the serological marks are the HBsAg, that is the first marker to appear in the course of infection. The anti-HBc IgM is a marker of recent infection. The anti-HBc IgG indicates prior contact with the virus. HBeAg indicates viral replication. Anti-HBe appears after HBeAg disappearance. And finally, anti-HBs appears after HBV recovery or after vaccination. These serological markers, together with biochemical tests and molecular tests, are important to define the laboratorial hepatitis B diagnosis, and also to characterize the course of chronic infection and to evaluate the effects of antiviral drugs on virus replication. For laboratorial hepatitis B diagnosis is commonly used serum and plasma samples, but it is of difficult execution in children, elderly people, and people with difficult venous access. So the collection of saliva could be an alternative in these cases, being cheaper, less invasive, and less painful when compared to blood collection. So it is increasingly used for hepatitis B virus detection and other viral infectious disease. So, since 1974, some authors are describing the use of detection of virological HBV markers in saliva samples. So, Cameron and Dane, in 1974, aimed to detect HBsAg in saliva and semen samples of chronic HBV men. As a result, they detect 18 of 24 samples that were HBsAg positive, but the amount of this marker in serum samples was low. In 1975, Irwin and colleagues had the same object to detect HBsAg in urine, saliva, stool, seminal fluid, and menstrual blood. As a result, 13 of 86 samples were HBsAg positive in saliva samples. And all the authors of this study agreed that saliva was considered infectious. But till today, no author could prove this. Recently, in 2005, other authors tried to validate the the detection of HBsAg in oral fluid sample using a commercial collector, and evaluated parameters of the reaction, such as sample volume, incubation time, and temperature, obtaining 90.7% of sensibility. Another study from our group of Brazil in 2011 had the same objective to evaluate the applicability of a modified ELISA for HBsAg detection using whole saliva and oral fluid collected with salivette device, and evaluated the following parameters: type of buffer, sample volume, and incubation time, and they obtained a similar sensibility. Back to 2005, other authors tried to verify a correlation between the HBV DNA measurements in serum, saliva, and urine of chronic HBV patients, using saliva collected by asking subjects to dribble into a sterile plastic pot, and using an in-house real-time PCR. As a result, they observed a high correlation of HBV viral load between serum and saliva. And in 2006, other authors could prove that the HBV DNA titer corresponded to the HBeAg positivity in serum samples, being the HBV DNA amount 2 logs higher in HBeAg samples. And also, they could prove that high levels of HBV DNA in serum correspond to HBV positivity in saliva samples. Hence, we became interested in comparing molecular methods for hepatitis B virus DNA detection from oral fluid samples. This study was published on Journal of Medical Microbiology in 2012, and I will present it to you. We collected samples of oral fluid from 20 healthy adult volunteers that were in absence of HBsAg and anti-HBC markers, using the collector salivette. With this collector, we put a cotton into the mouth of the patient for 2 minutes, and then we transfer it back to the tube and add 1 ml of PBS buffer, centrifugate, and after that, we pull these samples and dilute it. The pool of oral fluid was divided in 2 groups, in 2 aliquots. The first one was used as negative control, and another one was sterilely diluted, using a commercial sample, OptiQuant from Life Technologies, that contains 20 million copies of HBV DNA per ml. So, in the first tube of the serial dilution, we had 2 million copies of HBV DNA per ml, and in the ninth sample, we had 0.02 copies of HBV DNA per ml. For viral DNA extraction, we evaluated 4 commercial kits that had the same principle of silicon membrane columns, and were as following: saliva DNA isolation kit from Nargin, RTP DNA RNA virus mini kit from Invitec, high pure PCR template kit from Roche, and Kayam DNA virus mini kit from Qiagen. Since in saliva samples there is a low amount of DNA, we increased the initial sample volume of these kits and decreased the elution volume. We evaluated 3 in-house qualitative PCR for detection of the HBV DNA. The first one is a salmon acid PCR for pre-SS gene that amplifies 1100 base pairs. The second one was an acid PCR for the same region that amplifies 502 base pairs. And finally, one round PCR for core gene that has a product of 441 base pairs. First, we optimized the salmon acid PCR using Qiagen DNAs and evaluated DNA volume, annealing temperature of primes, number of amplification cycles, DNA extension time, and platinum-Taq DNA polymerase concentration. And all these parameters were used for the optimization of nested PCR. We also evaluated the cost analysis of the extraction methods, evaluating the costs of test kit, materials, and reagents based on prices from 2010, from July to December 2010. And we didn't include laboratory personnel salaries, equipment, and laboratory overhead costs. During the optimization, these following parameters were chosen for first and second round of semi-nested PCR, and the protocol, without the modifications, had a sensibility of, here, 2,000 copies of HBV DNA per ml. When we evaluated the parameters I previously described, we increased the sensibility of the method for oral fluid for 20 copies of HBV DNA per ml. For nested PCR, the same reaction conditions for semi-nested PCR were used, although we substituted a primer in the second round of amplification. And we also assessed the sensibility of 20 copies for oral fluid samples. Comparing the extraction methods with the semi-nested PCR, we could observe that Roche and Norgren kits amplifies 2,000 copies of HBV DNA per ml, while QIAGEN was the best one that detected 20 copies. Comparing the extraction methods with nested PCR, Norgren detected 2 million copies, 20,000, sorry, copies of HBV DNA per ml, while Invitec and QIAGEN were the best one, detecting 20 copies. Evaluating one-round PCR with the extraction methods, Norgren was also considered the worst, while Invitec was the best one. With this protocol was chosen to be used for oral fluid samples in our laboratory, one-round PCR, together with Invitec, because it has a good sensibility and we didn't observe smeared bands or unspecific bands in the agarose gel. So, according to the cost analysis, Norgren was considered the most expensive, while the kit from Roche was the cheapest. Nowadays, we are evaluating oral fluid collectors for HBV DNA detection, using 121 samples from 121 samples, and we are evaluating 4 collection devices. The first one is by spontaneous salivation. With this collection, we ask the patients to spit into this sterile plastic pot. The second method is by salivation, as I previously described. And we are also using DNSL. With this collection, this device is rubbed in the mouth of the patient, and then we ask the patient, to put this liquid in the mouth and switch around for 10 seconds, and then transfer the liquid back to the tube. And finally, the fourth collection device is by FTA cards. With this swab, we rub the mouth of the patient, and then we press the swab against the FTA card. We are also evaluating the parameters we evaluated before of concentration of platinum-Taq, DNA polymerase, volume of input DNA, and annealing temperature of primes. And our results, previously described results, from the 79 HBsAg patients, all of them had DNA detected by the commercial kit of PCR. And for oral fluid samples, we increased the volume of input DNA and the concentration of platinum-Taq DNA polymerase and the annealing temperature of primers. So, from the 79 positive samples collected with salivage, 14 had HBV DNA detected, and from the 79 collected with FTA card, 15 had HBV DNA detected. We could observed that only samples with high viral load in serum samples, in paired serum samples, could be detected using these collectors. Using DNA cell and whole saliva, we couldn't test yet all the samples, so we don't have a result. To conclude, all of detection methods of PCR can be used to detect HBV DNA from oral fluid samples, but the results obtained with one-round PCR and the kit from Invitec suggest that this combination is the best choice for HBV DNA detection in this kind of samples. Evaluating the detection of HBV DNA in oral fluid samples obtained by different collection devices, we demonstrate that only high-loaded serum samples can be detected using salivette and FTA card. Oral fluid samples provide a non-invasive method of detecting HBV DNA that can be used for children and people with difficult venous access, such as the elderly ones. The optimization of methods for HBV DNA detection is very important for improving the access to diagnosis in these populations. I would like to thank the viral hepatitis laboratory team that is in this picture, the volunteers who donated oral fluid samples, and also the financial support of FAPERJ, CNPq, and Fiocruz. Thank you for your attention.